twice the population size, because mutations accumulated both along this lineage and along this lineage. So that means that we immediately get the population size. Moreover, usually we have more than 2 individuals. We have larger samples than just 2 individuals. So, for example, we can have a sample of size n, of size 5. Okay? So, if there are 3 individuals, so for 2 individuals, we still have the population size n, but now we have different sample sizes. For 2 individuals, we have this coalescent process proceeding at the rate of 1 over n. What about 3 individuals? What will be the rate of 3 individuals? What's the probability that 3 individuals coalesce exactly 1 generation ago? Not all the lineages coalesce, but there is a coalescent event. There is some coalescent event among a sample of 3 individuals. So some one of them coalesces 1 generation ago. What's the probability of that? Well, there are several ways in which 3 individuals can experience a coalescence event, right? There are individuals 1, 2, and 3. So the first coalescence event that we see as we go backward in time can be either between individuals 1 and 2, between individuals 2 and 3, or between individuals 1 and 3. We cannot tell a priori which one of those will happen, right? And each of those happens with this rate, with this probability of 1 over n. So the probability of one of those events happening is just 3 times that. Okay. So for the sample of size, for the sample of, I don't know, of any size k, the coalescent rate will be this times the number of combinations that's m, let's just keep it m. We don't need the key, let's keep it m. The combinations of n choose 2, and for each such combination, this is multiplied by this rate of 1 over n. Okay? So here, if we have a sample size of 5, this will be what? 10 times 1 over n. And then we might ask, well, okay, so now we have the time, the estimate for the time till the first coalescent event. That's straightforward. Now, we can ask, okay, what is the time to the most recent common ancestor of the entire sample that we're analyzing? In this case, the sample size is 5, and that time is just the sum of the timings between all the sequential coalescence events. So first of all, some 2 individuals from our sample have to coalesce with each other. Then 2 of the remaining individuals have to coalesce. Then 2 of the 3 remaining individuals have to coalesce. Then 2 of the last remaining 2. So then the both remaining lineages have to coalesce with each other, and then we're done. So this will be the most recent common ancestor for the sample, and then the time to that will be just the sum. The expected time to that will be just the sum of the expected times to coalescence events of these thingies. So the sum of the mean of the sum is the sum of the means. And we can write it out, and it turns out that we just saw that for 2 individuals, the time to coalescence is twice is similar to the population size, right? Is n. Well, so this is the value for the entire sample. It is 2n times this value 1 minus 1 over n. So if we have a large sample, if n is large, then, for example, we have an infinitely large sample, n can be very large. So then this converges to just 2n. This is sort of curious because, so if we go back in time, it turns out that we can have a very large sample, and then these guys coalesce with each other. Most of them coalesce with each other rather rapidly, but then the population just spends half of the time waiting for the last common ancestor, actually waiting for the last two the remaining lineages to coalesce. Okay, so this T2 is usually as long as the sum of all other coalescence steps. So we have derived all that sort of on the back of the envelope using just the logic and the idea that this is a Rice-Fisher process. So if this constitutes a Rice-Fisher process, if our population is a Rice-Fisher population, then we can be sure that we can tell in advance what the characteristics of this process will be, and basically what the shape of this coalescent tree or this coalescent will be. We can give general predictions such as this last time will be as long as all the remaining times. And these trees are constructible from the data. So we can go to the data and ask what the actual shape of this tree is. And compare that to what we expect under the Rice-Fisher model. And then we can ask, well, how consistent is our data with the Rice-Fisher model? And if it's not consistent, then what is the model that explains our data better than the Rice-Fisher? Maybe our population size has changed with time. So that is something we actually are interested in, right? So again, so we do not, okay, so as a side note, we do not necessarily have to have all the samples from the last generation. Maybe sometimes we can have historical data, some samples from some previous generations. Nevertheless, we can construct the coalescent and look at the distribution or the branch lengths. So now assume that what we see is a shape of a tree that is very different from that expected under the Rice-Fisher model. We do not spend half of the time waiting for the last coalescence to happen. Instead, the last coalescent event happens unexpectedly rapidly. So the branches near the stem are short, and the branches near the leaves are disproportionately long. So that means the Rice-Fisher model is violated. That means the Rice-Fisher model doesn't work. Okay? So what could go wrong? Well, most obviously, probably the population size has been changing in time. So what we see is that the coalescence rate changes over the course of the coalescence process going back in time, right? Here, the coalescence rate is sort of low compared to these guys. And here, it is unexpectedly rapid compared to these guys. The fact that coalescence is rapid, it means that the population size is small. So it has to imply that the population size has been larger in recent times and smaller in more ancestral population. So it has to imply that the population size has changed. And then I'm going to ask, well, what's the law according to which this change is proceeded? And for example, we can try to feed an exponential model of population size change and ask which constant of that exponent characterizes the data best. So just by looking at the shape of the phylogeny, at the shape of the tree, we can tell, we can infer the basic parameters of the population history, in this case the population size dynamics. By contrast, so this is what we've discussed, by contrast, if the branches near the tips near the leaves are disproportionately short, and down, right? You were coalescing very rapidly in recent history, but you were coalescing slowly more and more ancestral populations. Again, this is something that you expect under constant population size under the Wright-Fisher model. So the length of this time is equal to the sum of all the other coalescent times. And if, for example, the population has been growing exponentially, you expect something completely different. You have to expect this different shape of the coalescent tree. People have tried to use considerations like that for actual phylogenetics and try to ask what are the distribution between the branching times of lineages on the evolutionary trees. Say we can take an evolutionary tree of all vertebrates and ask what is the distribution of branching times between the speciation events giving rise to different species. And people have tried to infer laws governing the speciation process from that. The problem is, of course, that while we do have a nice model, null model, predicting to us what the coalescent process should be in a simple population genetics scenario, we do not have a null model for evolution. We do not for microevolution. We do not have an a priori expectation for what the rate of speciation should be. Nevertheless, you can ask, for example, well, is the data consistent with a uniform rate of speciation in time? And then you say, no, it's not. There are things like the Cambrian explosion in which the rate of branching is much faster than in other periods of time. So then you can ask, well, what does the distribution look like? And so forth. People are doing that. That's a little bit of a soft science to me because we do not actually have null predictions from any first principles of what the rate of macroevolution should be. So I have sort of been discussing the case in which you know a priori the shape of the coalescent. But so if you have a large sample, for example, of many individuals, then you can do this coalescence analysis and do all these nice things. If you just have a sample of two genotypes, there's not much that you can do, right? You can just infer one parameter, you just compare two genotypes and infer one parameter and count how divergent they are. Well, they will not be, that just gives you one estimate of n. Oh, by the way, let's go back here for a second. So do you remember this result in the genetic drift under which we have shown that the pi, we went into some trouble doing this drift mutation model, and there we have shown that the pi is close to 2 effective population size times the mutation rate, right? So we have been writing out all the equations and finding equilibria, and this is what we got at the end. Well, in the coalescent reasoning, this is something that we get immediately if we just look at this picture, right? Because, so what is this? So these are two individuals randomly sampled from the population. And the difference between them is exactly what pi is about. And what is it? Well, we just asked what's the mean coalescent time between these individuals. And here it is 2 times n, 2 times the population size. And that's how many mutations have they accumulated over this time course. Well, it's just the time separating them times the mutation rate, which is mu. So we get this equation just immediately just by looking at this figure. This sort of hints at the power that the coalescent methods have in population genetics. For some things, they're extremely powerful, much more powerful than the direct analysis of equilibrium and such that we've been dealing with previously. So there's not much that we can do if we just have a sample of 2 individuals, right? We can just get one estimate of one parameter n from it in this way. Or is there? Well, if these are sexual individuals, if these are sexual populations, they constitute sexual populations. There is more than we can do, because the different segments of each genome have undergone recombination, and in essence, different segments of the genome can have different population histories. They can be unlinked. And then, for example, we have two, I don't know, humans, we can, two human genotypes, we can ask what is the coalescence time for these two individuals, not for the genotype as a whole, but for each individual segment of this genotype. And in general, these segments will differ in their coalescence times. For some segments, we have had a common ancestor just a few generations ago, because we both inherited this from our very recent common ancestor. But for some other segments, we could have inherited them from our much more distant common ancestor. And then, in theory, if we can get the estimate of the mutation rate, or the coalescence time, right, or the difference, and therefore coalescence time, for each genomic segment, we can ask what is the distribution of these coalescence times over the whole genome, and that sort of should tell us something about the population history just by looking at two individuals. So there is an actual method for that, actually a bunch of methods, but this one is most popular one is something called PSMC, that's pairwise sequential Markovian coalescent. You do not have to remember that, although you will be using it in the computer lab. That's right? Yeah, that's the method that we'll be using in the computer lab. So the way it works is that you assume that the genome is you assume a Markov process, hidden Markov process along the genome, and for each region or segment of the genome, you ask what is the rate at which you observe the differences between two genotypes. So it's a pairwise method, so it just takes as input two sequences of two genotypes, right? And all that you have is the just distribution of these red dots, which are the positions of differences between these two genotypes. And then, so there are some lumps, some regions in the genome in which these differences are very densely packed. There are quite a lot of them, and that means that in this region, the last common ancestor has lived a long time ago. So for this region, the coalescence event has happened a long time ago, so that's a large time to most recent common ancestor. And for some regions can be almost homozygous, almost identical between two individuals, and that means that the common ancestor has existed just a few generations ago. Then you can pack all of that, so integrate over the genome, and what you get is the distribution. Okay, so here for each genome coordinate, we have this time to most recent common ancestor, and then of course you can do the bootstrapping of some sort to get the confidence bands around your mean to get an idea of how sure you are. And then what you can do is use just this information coming from just these two genomes to understand the whole distribution of coalescence types. And once you have the whole distribution of coalescence types, you can ask how similar is it to that expected under the Wright-Fisher model. And if it's not similar, then what does it tell you about the population size dynamics in the past? So just by looking at two genotypes, you can infer the population history. And we're diploids, and many things around us are also diploid. So that means that you can just get the DNA from a single individual, and just by looking at a single individual, understand the population history of that. And it does not even have to be a currently living individual. This is a mammoth bone that people got from Wrangell Island. So it's a mammoth that lived actually pretty recently. There was a small population of actually pygmy mammoths there that survived up to just about 4 or even 3,000 years ago. So all the researchers had what a sample of DNA from these mammoth tooth, and they processed it as I've just described, and they have got a graph of a plot of the past population history of this population just from the DNA from the single bone. So the way to read these plots is as follows. You go back in time. So in coalescence, it's always sort of perverse because you always have to think back in time. As you go back in time, this becomes larger and larger times yeah, that's right. So this is recent past. So this is time in units of 2 mu tau, right? You don't know the mutation rate. If you do, you can recalculate that in years. So you go back in time and ask what the population size in these units of the mean, if I calculate the size here, it doesn't matter, what the relative population size has been at this point of time. And what we see is that the population was originally, a long time ago, it was pretty large, and then it went into sort of a decline at some time point, and then it increased back, and then it declined not long before these mammoths actually went extinct. So these can tell you sort of that maybe, okay, so this is about 10,000 years ago, this is the end of the glacial period, so maybe that has to do with the fact that the glacial period has ended and the mammoths didn't do that well in the interglacial that they did before. And then you can have a sample, so here they had an independent sample of a different mammoths of a different age, about 40,000 years ago, from a location in Oymacon, and you see that even though it's a completely different individual, the data matches pretty nicely. Here the plot ends at this time 44,000 years ago, which is when these both have been estimated. So I think that's true. People have done that for other species. For example, in this case, there are these are the different bird species, and they have used that. Somehow the colors don't show all that well in this projector, but so if you look at the slides online, you will see that there's like these gray rectangles going to the left from the 10 to the 5th mark, and that corresponds to the last glacial period. So the decline of all these species has largely corresponded with the onset of the last glacial period about 100,000 years ago, and has continued till about 10,000 years ago, till the end of this glacial period. So even so, for some of these species that are threatened, their populations have already declined long before they were attacked by humans. Not to mean that humans did not contribute to that decline, they certainly did. More species like that. So that's what we can do with things like animals and birds, so the mammals and birds, for which we have the recombination events along the genome. But for viruses we don't, but normally we don't. But still we can get the shape of this tree if we have a sufficiently large sample and ask what the changes of the population size of this virus have been. And I have told you yesterday about this fundamental parameter in epidemiology called R0, which is the rate of exponential increase in the size of the population, or decrease if it is lower than 1. So this is something people have cared a lot epidemiologically in the pre-genomic time, and now we can estimate that by just looking at the coalescent tree, right? So you have the coalescent tree, and if you see a tree such as this, a tree such as this, it means that your population is growing. For viruses, it means that you have an epidemic increasing, and you, by the shape of this curve that you feed to this data, you can estimate R0. So if you have an outbreak, like Ebola outbreak, somewhere in the Africa, right, and you just discovered you have this outbreak, you don't have anything, you don't know anything, you don't know for how long it has been proceeding, you have people dying, but just you can go and sequence the virus, but not much beyond that. You can still sequence the virus and do the coalescent and ask, well, is your epidemic on the rise or is it going down? Okay. I think that's pretty cool. So this is the, so for some of you may remember the swine flu scare of 2009. This was the year in which there was the new subtype of H1N1 influenza arising in the human populations, which we got, which was a recombinant subtype that we got from domestic pigs, and people were extremely concerned that this is a start of a new bad pandemic because it was spreading among the humans pretty rapidly. It turned out ultimately that it was a start of a bad pandemic, but the virulence and the mortality associated with this H1N1 subtype was similar to that associated with the previous seasonal flu, so it didn't make much of a difference. However, the data back then was too scarce to tell everything, to tell something about how bad it was, and there was some early data showing that people who were dying of it tended to be of younger age than those usually dying from seasonal flu, which everybody sort of got very scared about because that was very reminiscent of the Spanish flu. And so the first viral strains, I believe, were the first instances of this new infection arising were detected in March of 2009. And this is a paper in Nature that has been published, I believe, in July 2009, so 3 months after the first strains were detected. And so this is a very early, very preliminary analysis that people did of the rate of spread. But this is exactly one of the things that they did was exactly the thing that I told you about. They tried to estimate the rate at which the population has been growing, the rate at which the infection has been spreading, using a bunch of different methods, but one of the methods was exactly this coalescent-like approach. And they saw sort of, so the variance was, the error was large, but they saw that it is significantly larger than 1, so they did see that the pandemic was on the rise. So that's the first thing you do in an outbreak, right? You sequence some genomes and do the coalescence. Okay, let's start talking about the so I'm wondering if we should start a large new topic, or we should stop for the journal club now. What do you think? Sorry? How many? I have 20 more minutes. Okay. So the fourth thing that we might be interested in, and the fourth thing that we might want to learn from the about an virus is selection, actually, on it. And there are a bunch of reasons why we want to ask how selection is distributed. For example, we might want to design a vaccine, and we want or drug, and we probably want the drug to target the positions of the protein, the amino acids of the protein that are conserved, right? Because changing these amino acids would be costly to a virus, and if we target them, then it will be hard for the virus to acquire drug avoidance. By contrast, if we target some amino acids that are very flexible and easily replaced in evolution, probably the virus will just substitute them with something else without much cost to itself, acquiring drug resistance. So that's one reason. Maybe we want to design with C. So the viruses are iconic for the pathogens, are iconic for the level and the high intensity of adaptation by positive selection, that way that they acquire, and the way in which people usually think about that, that's something we have already hinted at throughout, is this something called the Red Queen scenario. So this is called, of course, after the Red Queen in Lewis Carroll's Alice in Wonderland, who said that Alice behind the mirror glass, not in Wonderland, the second book. She's saying that here it takes, in this country, it takes all the running you can do to keep in the same place. That's exactly what the viruses apparently have to do. They have to evolve constantly in order to avoid the constantly changing fitness landscape imposed by the host immune system. And okay, so the simplest thing you can do is to just to map the positively selected sites and to map the negatively selected sites, and that already tells you something, right? You may want to design drugs targeting so this is the envelope proteins of HIV, and this is the surface hemagglutinin proteins of influenza, and here you might want to target the blue negatively selected sites, but not the yellow positively selected ones. So as a side note, we do see that the virus is evolving very rapidly in real time. The viruses that attack us humans are changing over the course of over the time scale at which we have been studying them, and these changes, we do see that they make them more adaptive, more well adapted to human, to what we humans try to fight them, like our immune system. So here's an example of one such adaptation. So different human populations change. Okay, so there are some genes that we have encoding some proteins that we have that provide partial protection against HIV. We have discussed the major histocompatibility complex, which is in human called human leukocyte antigen, and we humans host, as we also have discussed, a large variation in these antigens. There's balance and selection acting on the diversity of these antigens, and there are different humans have different haplotypes of them. And different forms, different alleles of these genes have different efficiency against the HIV wild type, against the HIV that has infected humans in the first place. And different human populations have different prevalence of different HLA variants. But also different human populations have different prevalence of different HIV subtypes that attack them. And here's the analysis in which people saw that these two things turn out to be correlated. So on the horizontal axis, this is the frequency of a particular variant, HLA-B51, which gives partial resistance against a certain HIV allele. And on the vertical axis is for the exact same population, the frequency of an HIV variant in that population, that is an escape variant from this HLA variant that the humans in this population have. So we have this Kumamoto population, for example, in which the HLA-B variant is rather frequent. It is has about 25% prevalence, and in that population, the majority of the viral strains have the escape mutation. The geographic distribution of HIV mirrors that of human genotypes that are efficient against these HIV subtypes. So the selection, in essence, against certain HIV subtypes or subtypes is local. And this is a dynamic process. This changes in time. About 10 or 15 years ago, this subtype B51, this B51 allele has provided partial protection to many of the Japanese. Japanese were better protected against HIV than, say, Europeans or North Americans, exactly because of the high prevalence of this HLA gene in them. Not anymore, because this escape mutation has increased in frequency, and so there the incidence, not the incidence, but the virulence of the virus is similar to that elsewhere. And you can have the same plot for different other alleles of the gene, and each time you see the same thing. The populations in which a particular allele providing partial protection with a high frequency, in that population the escape variant was also a high frequency, which sort of is bad. Okay, so you probably have heard this story before, but this is this has been in the news several times over this year. So some of the humans have inherent protection against HIV infection due to the fact that they have this delta 32 mutation in their CCR5 receptors, which is delta 32. That just means that 32 amino acids, a chunk of 32 amino acids is cut out of this protein, and this is the receptor that the HIV particle actually uses, one of the receptors that HIV uses in about half of the cases for entry into the cell. So if an individual is homozygous with respect to this mutation, he or she is apparently, in as much as we know, completely protected from HIV of this type, of the HIV which uses this co-receptor for entrance into the cell, although not against the HIV of the other frequent type, which is called X5. And so if you're homozygous, you're pretty much protected. If you're heterozygous, then you are usually what's called long-term non-progressor. That means that even if you get HIV positive, you do not usually develop symptoms of AIDS for a long time, even without any kind of treatment. And so this mutation is rather frequent in human population. It looks like a loss of function mutation, right? Because it disrupts the function of the protein, and it's very interesting to know whether it is costly in some our respect, whether it actually handicaps the immune system for some other functions. That's something we do not yet understand. People have been trying to find the downside of this mutation, but as far as I know, none has been shown so far. Still, this mutation does not have a 100% frequency, but it has a substantially large frequency. In about, especially in Western Europe, it has the frequency of the order of 10%. No, it's not that frequent in the rest of the world, and so people have been asking, well, why this distribution? And people have thought initially that this mutation has been a defense against bubonic plague, because the timing of the spread of this mutation has seemed to coincide with, say, like 15th century, and it is, as expected for the bubonic plague, mostly confined to Western Europe. Nevertheless, when people have sequenced the fossil skeletons of individuals that lived in Europe in the Bronze Age from Italy and Germany, say like 3,000 to 5,000 years ago, they also found the same mutation with the same frequency. So that means that that mutation has already been spread in Europe 5,000 years ago. It was not a response to bubonic plague. It could have been a response to some earlier epidemic or pandemic that has spread through Europe back in that time that we have no idea of, and we will probably never find out anything about. So sometimes you're going to have heard about the Berlin patient, the only, actually up till today, literally today, I'll tell you in a second, he was the only person known to be cured of HIV. He was the person who had the transplant of bone marrow for his lymphoma, which he had in addition to HIV, from a patient, from a donor, a healthy donor, who had this CCR5 delta 32 mutation, and as a result, some of his immune system cells acquired this mutation, apparently enough of these cells to rid him entirely of the detectable levels of HIV. Now he is off drugs and off the HIV antiretrovirals, and he does not have detectable level of HIV relapsing back into his blood. While pretty much anyone else, if he gets off drugs, then he gets relapse newly detectable levels of the virus. Literally today in Nature, a paper is published on a second instance of the same mechanism. So a second person has had no detectable levels of HIV after bone marrow transplant. He used to be an HIV positive patient. He also had Hodgkin's lymphoma and had a bone marrow transplanted into him to cure, as a cure for the Hodgkin's lymphoma. The first Berlin patient was put on this very extreme immunosuppressive therapy, which was the standard treatment back in late 90s or early 2000s when that was done. So that was a pretty extreme immunosuppression. He had to be led into an artificial coma for some time. He nearly died. This second patient had experienced much softer immune immunosuppression regime, and so basically this tells us that you do not have to nearly die to get cured of HIV via this mechanism. Still, of course, this is not something that you would recommend to an HIV-positive patient, right? Because a bone marrow transplant is an extremely risky procedure, and now we do have antiretrovirals that allow you to survive for tens of years. Basically, your expected lifetime is similar to that of a healthy individual, even if you're HIV positive. So the current generation HIV drugs allow you to stay disease-free for tens of years, and also they prevent, as has been recently shown, transmission of HIV to others, including to your own children, for the most part. So that's not something that you would do normally, but still, so that's a nice instance of that's obviously, that's an instance of a variation of the human population that has to have some selective ground, but we do not really know what it is. Another thing in the use that had to do with this CCR5 gene in this year was something you've probably heard about, this Chinese guy who did the CRISPR gene replacement on the human embryos. So this is exactly the gene that the mutation that he has incorporated into these two twin girls. This is the mutation that he put there with the aim of making them disease-free, allowing them not to acquire the disease from their parents. And that's of course like an experiment, a treatment that has been heavily criticized because there, among other things, because there are other ways to prevent transmission of a virus from the parent to the offspring. Okay, let's stop here, and then next week we'll start where we left off and discuss how the HIV actually evolves not on the population level but within individuals. So next week we'll finish the evolutionary medicine part, and then we'll have some time left to talk about the quantitative genetics. Other journal topic, right?